And you probably heard, okay. Those of you who are already in the meeting would have heard Daphne say that we heard Professor Allen-Vercoe speak at the CURAC meeting held in Guelph about 1.5 years ago. And we all thought that she gave a terrific speech, terrific talk. And I thought she would make a great speaker for senior college. And so 1.5 years later, here we are by the magic of Zoom. A little background about Emma. She did her undergraduate degree in biochemistry, and I have to say that was an excellent choice, at Royal Holloway and Bedford New College. So just as an aside, if you do not know Royal Holloway and Bedford New College, it's in a beautiful campus with wonderful Victorian architecture, located just outside of London, not far from Windsor. And sometime, if we can ever travel again or in London, it's well worth a visit to have a look at it. Anyways, Emma indicated to me when we set this talk up that somewhere around about the 3rd year of her study, she realized that her passion lay in the field of microbiology and not biochemistry. So she switched fields and completed her PhD in molecular microbiology at the Open University. Following her PhD, she did postdoctoral research for a couple of years at the Center for Applied Microbiology and Research at Porton Down, before making the move across the ocean to take up another postdoc at the University of Calgary. Postdoc at the University of Calgary transitioned into a faculty position at the same university, and she held that position for 10 years before moving east to the University of Guelph, where she is currently professor and Canadian Research Chair in the Department of Molecular and Cellular Biology. Her research interests have included analysis of the role played by pathogenic strains of E. coli and Salmonella in infections in both man and animals. Research which has the potential of reducing the use of antibiotics in intensively farmed animals. In other work, she has studied the relationship between oral microbes and inflammatory bowel disease and colorectal cancers. Presently, the main focus of her lab is the modeling of the human gut microbiota using an in vitro system. Culturing the microorganisms of the human gut has long proven a difficult nut to crack. And really, given the source of the bugs, who would want to do it? But she does do it, and she's largely succeeded where others have failed. Indeed, her lab is among a small group of laboratories worldwide that is equipped to carry out studies of the microorganisms that populate the human gut. Her research has recently led to the founding with 3 other collaborators of a biotech company called New Biota, the current primary aim of which is to provide a treatment for recurrent Clostridium difficile infections, an increasing problem in our hospitals and long-term care facilities. So this is pretty important research. Along the way, Emma has, of course, published numerous articles and been the recipient of several awards, including the John Evans Leader Fund Award, the YWCA Women of Distinction Award, and the Partners in Research Ambassador Award, among others. She has been a committee member of the Human Microbiome Project and represents Canada as part of the International Human Microbiome Standards Group. So just one final note before I pass the virtual mic over to Emma for her talk. I mentioned that she did her undergraduate degree in biochemistry at Royal Holloway College. As it happens, I have a very good friend and colleague in the biochemistry department at Royal Holloway. And when I learned this fact about Emma, I asked my colleague, as one does, if he remembered her, and at the same time copied to him a TED lecture that she had given several years ago to help jog his memory. He replied that he indeed did remember Emma, and as he recalled, she was more interested in microbiology than biochemistry. So apparently, even as a somewhat disinterested undergrad, she made a lasting impression. And on the basis of the TED Talk, my colleague commented that she appeared to be a very good speaker. And we are all now about to learn that for ourselves. So without any further delay, Emma, I pass the mic over to you.
Oh, thank you so much. That was really fun to hear. I mean, I honestly didn't think that that he would remember me. I thought it would be quite an interesting thing. I might have to reach out to him and see how he's doing. So thank you very much, and thank you all for being here and putting up with Zoom. I think this is the new way of doing things, and so I will do my best to make this an engaging lecture, despite the fact that I'm not in the room with you. So first of all, I'm going to just try and make sure I can share my screen, and then we can get going. Okay, so hopefully you can all see my screen. And somebody give me a thumbs up. Jim, could you give me a thumbs up? That's okay.
Yes.
Okay, good. I always like to check because you never know. All right, so today I'm going to talk to you about a little bit of the research in the lab. I've deliberately kept this a little not too science heavy, because I think that there is so much research that's going on in this area that I could very quickly get into rabbit holes that are very kind of specific and perhaps less interesting to some of you. So what I've tried to do is to put together a talk that is more about a sort of a philosophical question of, can we use bugs as drugs? So can we use the gut microbiome, the microbes that live in the gut, to create, like, if you like, the next generation of probiotics, although you'll kind of see later on in the talk that we don't call them probiotics for some very good reasons. And hopefully in the lead up to that, I'm going to explain why this is such an important area of research and why it's all of a sudden, big news. And I think this, of all the fields in microbiology right now, this is probably one of the hottest fields. So I'm really pleased that I kind of joined it during, in the very beginning, when it was when it first started. And I've seen the microbiome field from the middle of the last decade, so around 2005 to now, and it really is a very, very different field, to the point where I'm having to learn so much more than I ever had to learn when I was an undergraduate. So I do feel sorry for some of my students these days. So I'm going to start with an introduction, by way of introduction, a question. And the question is, what is your gut microbiome and why is it so important? And I just wanted to illustrate this with some beautiful images that a colleague of mine took from, these are scanning electron micrographs from, actually, it's a pretty gross experiment, but for a few years ago, we did a Nature of Things documentary, which was a sort of a myth-busting episode with Jennifer Gardy, where one of the myths that we were trying to bust, if you like, was that when you eat some sweet corn, the sweet corn passes through undigested to the other end, and what's going on there. So what we did is Jen and Gardy ate a lot of sweet corn, gave us a sample, and we dug through the sweet corn to get dug through the poop to get the sweet corn, and then basically imaged it with scanning electron microscopy to show that the contents of the kernel of corn were in fact being digested just very nicely by these microbes, these gut microbes. And so as a result of that, we got some beautiful images that were taken here at the University of Guelph. We've got a very nice imaging facility here. And so I thought I'd share a few of these with you. So they'll kind of pop up every now and again in the talk as I go on. So you can sort of see from this image, you can sort of see the sort of chunky stuff is obviously just the sort of undigested starch from the kernel of corn, but you can very clearly see the microbes. And I also hope that you can see some sort of like tendrils coming out of these microbes, which are probably some of the sort of fimbriae, or the pili, as we call them, little hairs that kind of join these cells together as an ecosystem. And the reason that I point that out is because we tend to think as microbiologists, as microbes as individuals, and unless you're a microbial ecologist, you don't really have a sense that these microbes work together as an entire community, and they don't really have their own identity in that kind of sense. What they do is very altruistic towards other microbes for the most part, in these sort of healthy ecosystems. And so it's really important to appreciate that the gut microbiome in particular is a very, very important ecosystem, and that it's made up of many, many different bacterial species, but that they all work together. And the work that's been done in microbiology so far has tended to sort of put everything into silos, and so we're trying to sort of bring that together and bring a microbial ecology perspective to the field. So probably the other important thing, if you take nothing else away from this talk, then this is this point, is the fact that we are not human. And we tend to think that we're human, and we tend to, assume that that's all we are. Okay, now of course we're human, but we're not just human, is the point that I'm trying to get across. We're actually superorganisms. All animals are. The superorganisms of human and microbial cells. And we've completely ignored the microbial side of things for the longest time, and particularly in human medicine. It turns out that these microbial cells are probably one of the most important parts of us, and that we exist in an extremely delicate equilibrium with them. And so the fact that we've ignored them has actually been very detrimental, as you're going to see in a minute. So how human are we? Well, a few years ago, microbiologists have always been asking this question. And probably when I first started in this field, the estimate was that we are about, we're only 10% human. I think some of you have probably heard that. It's a very, it's based on some flawed mathematics. And so a few years ago, Sender, Fuchs and Milo did a study where they actually did do a much more careful count up of the number of cells that were present in a typical healthy adult, and the number of human cells that were present. Obviously, these are just based on averages, but this is based on a much more clear count of what was there. And when they did that, they found that actually we're not just 10% human. We're actually around equivalent. So there's about 1 part human to about 1.3 parts bacteria when we're talking about the numbers of cells. So we're just talking about the cellular life here, and we're just talking about the bacteria. Now, of course, in the human gut microbiome, there are other types of microbial life as well. And these have also been ignored for the most part to our detriment. I think, in my lab, we're starting to look at some of those, but for the most part, the research has been done looking at the bacteria. And the bacteria are the most prevalent form of cellular life in the human gut. Now, the question then, of course, why don't we look like bacteria? Well, I think most people here would realize that it's because bacterial cells are actually far smaller than human cells, on average about 1 in 100 to 1 in 1,000th of the size. Now, each gram of feces contains around 10 to the 11 bacterial cells. It's an extremely dense ecosystem. So every time you take a bowel movement, that's about 10 trillion cells that you are flushing down the toilet. So if you ever feel like you're more human after you've had a bowel movement, the reason for that is because you are. Because actually at that point, you've redressed the balance a little bit, and it takes a little while for those microbes to sort of re-establish themselves. So another pretty image just to sort of show you. This again is looking at this this sweet corn experiment, but just to this I think is an even nicer picture because it's showing the diversity of bacteria there. And I think the problem is that when we think about bacteria, we just think of them as sort of some nebulous thing, and that they all look the same because they're all single cells. And they're just kind of blobs. They don't really do anything. They're not like animals. The thing about bacteria compared to animals or eukaryotic cells, I should say, is that eukaryotic cells have many different forms. I mean, we are made of eukaryotic cells, and so are whales, and so are fungi, and all of those kinds of things, whereas bacteria are prokaryotic cells, and their form is very kind of limited to these sort of what look to us like amorphous blobs. But metabolically, bacteria and other prokaryotes have had far longer to evolve than eukaryotic cells. And so as a result, their biochemistries are extremely clever and very advanced. And their metabolic repertoire, if you like, is far, far greater than what could be potentially done by a eukaryotic cell. And I also, I tend to sort of think about these bacterial cells, and I've been working with them for a while, that they also, if you look at them under these kinds of circumstances, they also have variation in their form. And I think hopefully this image sort of shows you that. So you've got that cell in the middle that looks like a peanut, and other things that look like they've got these sort of fuzzy coats and things like that. So there are differences in these cells. They're not all the same. And if you look at them and what they can produce, some of them will surprise you, I think, because some of them are quite quite a lot more, what's the word I'm looking for? Clever and sophisticated than we give them credit for, because we just tend to think of them as simple, single-celled organisms, but actually they have some very incredible behaviors, and especially when they are part of an ecosystem like this. So there's power in numbers. The other point, and I think that this is the biggest point, biggest take-home message of all, actually, for this talk, is that it turns out that biodiversity in the gut is important, just like it is in pretty much every other ecosystem on Earth, whether it's plant, the macro world or the micro world. And that results from this factor here. So we have the gut microbiome is usually, when it's healthy, is a very biodiverse environment. And when you have high biodiversity, you have high diversity of species, that gives rise to a robust ecosystem. And the reasons for that are many, and I'm not going to go into an ecology lecture right now, but just in very kind of broad terms, it's about balance, and it's about what we call functional redundancy. So in other words, you have a very high gene count collectively within an ecosystem. You have many genes which can do the same kinds of functions. And therefore, if you were to take one of the species away, there will be some other species in a diverse ecosystem that could step up and take on the role of the missing species. And outwardly, you wouldn't notice the net effect on the ecosystem would be very minor. And so you have resistance to damage. There's a threshold that you reach, but if you keep plucking things away, then eventually you're going to lose function, and then you will start to reach this kind of scenario where you have a low diversity of species. And I've pictured this here as a sort of this is actually Kananaskis National Park, near where I used to work at the University of Calgary. It's a very low diversity ecosystem naturally, just because of where it's placed with its geography and exposure to the elements and to UV lights very high up. And as a result, in terms of the biology there and the species there are not that many species in comparison to something like an Amazonian rainforest. And those kinds of ecosystems are very fragile, and that's sort of been I suppose, in a way, illustrated by the fact that the pine beetle has come across from British Columbia and started to decimate the white pines in the region. And those are a keystone species in that ecosystem, and that causes a problem and a breakdown of that ecosystem. So that kind of damage is a result of this naturally low diversity in the ecosystem. But in general, when you have a low diversity ecosystem, it means that that ecosystem can take fewer hits before it's really kind of decimated, and that can cause problems. So I've been speaking in terms of forests there, but as I mentioned, the macro sort of ecosystem and the rules that apply in ecology there are very similar to the rules that apply in a microbial ecosystem as well. So in terms of the human gut, so the bacterial community in your gut remains relatively stable from the time that you are weaned to the time that you reach old age. And we are just beginning to understand why. For a while I had this slide up and said, we have no idea why this is, but now we do understand. A lot of it has to do with our immune system, which of course is educated by our microbes early in life. And a lot of it has to do with early life exposures to microbes. And that's a very critical time point, a very critical window. Now, this talk isn't going to be about that critical window, but it is something to consider. That period of time from the age of 0 to the age of about 3 is when our ecosystems are really developing. And once they have developed, they tend to stay very kind of stay very similar through the rest of your life. And so that's really important. So if you're raised in an area or in an environment where you haven't had a lot of exposure to microbes, you're already setting yourself up for a lifetime of problems, because your ecosystem might not be as diverse as perhaps it should be. And the question about how much diversity is good, we don't know the answer to that. And it's probably very different for different individuals. But we are getting some clues now. So this is actually from a paper that came out last month, I believe. So this is based on a study, the twin study in the UK. And what this is doing is really showing us some interesting data from the point of view that in the past, the sequencing analysis was a very, very difficult thing to do. Now, that is no longer a barrier. The sequencing technology has really improved. And so the biggest problem that we had was that there is so much variation in people's microbiomes that it was very difficult to correlate anything to anything, just because everyone is different and you know, there's so many things you have to look at. The only way to really look at data sets like that is to look at huge data sets of people to look for trends. And now we're reaching that type sort of realm where we can actually do that. And so some very seminal papers have just come out in the last few months. It's been a very busy time for reading for me. But this one is particularly interesting because it illustrates a point quite well. So as I mentioned, everyone has a unique gut microbiome composition in terms of their species. And you can see that on the pie charts on the right-hand side of your screen here. So on the left-hand pie chart, that's the species composition. And on the right-hand side is the pathways composition. So in other words, what the actual microbes are, what genes they possess and what pathways, what biochemical pathways they can carry out. And so what you can see is that in the yellow here is strains or species or pathways which are detected in more than 75% of the sample. So you can see that in terms of species, there's very little shared across this population. And this was a group of twins. So this is a twin study. Looking at, I think I can't remember how many now, but quite a number of individuals, and what I hope you can also appreciate that on the right-hand side, when you're looking at pathways, those pathways are that sort of yellow chunk of the pie chart is much larger. So it's really not the species that's important, and unfortunately, that's what we have focused on for the sort of first half of this microbiome renaissance, if you like. We're so kind of fixated on bacterial species, and it really doesn't matter what the species are. It's what pathways they have to offer to a given ecosystem that's important. So what do our microbes do for us? Well, they do many things, and this is just a really small number of things, but perhaps some of the more important ones. So one of the things that they do is they regulate the immune system. They, in fact, educate the immune system quite importantly in the early period of life. That sort of tolerance that you gain as a result of that will dictate whether or not you have allergies later in life, for example, as well as autoimmune diseases and all sorts of other issues. They obviously help to extract energy from foods. I think that was sort of well recognized in the beginning, but it's one of the kind of one of the early things that we thought the microbiome did, and one of the only things we thought the microbiome did, and I don't think we could have been more wrong. They control potential pathogens. In the past, again, it was believed that they just control these potential pathogens because they crowd them out. Now we know from work done in my lab and others that a lot of the microbes that are present in the human gut actually produce metabolites and other compounds which actually control the virulence of potential pathogens that are coming in. For example, salmonella. So salmonella, obviously we know it causes a nasty kind of food poisoning, but it doesn't cause food poisoning in everybody. And the reason for that is because if you are lucky enough to have a diverse microbiome full of these kinds of microbes that make these particular metabolic products, what will happen is those metabolic products will permit the salmonella to carry on its journey through your gut as it's passing through, but it will prevent it from behaving in a pathogenic way. And so they're like a little bit like a police force. And so I think that that's a very important and understudied aspect of what the gut microbiome does. They make some essential metabolites, including vitamins and cofactors. They improve intestinal function. They remove toxins and carcinogens and many other things. And so really we can think of our gut microbiome as important to us as a liver. Really, it's a virtual organ. The fact that it isn't actually an organ that we can see in the way that we can see a liver means that it has been ignored, again, to our detriment for the longest time. Now, the big question in the field right now that I think we've pretty much answered, and I'm going to take you through it, is this one, has microbiome diversity, as I've told you, diversity is very important, has it been eroded in the Western world in particular? And this sort of comes from a sort of a thesis written by Blazer and Falco back in 2009, developed for over several years based on their own research at the time. And what they were suggesting was that that the microbiome is something that it might be with you for life, but it's easily damaged. And when it's easily damaged, when you damage it, maybe through eating junk food and sitting around on the sofa or taking pharmaceutical drugs, when you damage it and you lose those microbes, they become extinct from a microbiome. So they may not come back. And when that might be inconsequential, as I said, you know, your microbiome, if it's diverse enough, might be able to cope with a few hits. But the issue is that when you look at this over generations, there's a different story emerges. And on the left-hand side of your screen, you can see their actual sort of figure, which has become quite famous now in the field, where they were sort of predicting what might happen. And so here is just a sort of a representation of a conserved microbiome. So it starts off at about, you know, let's say it starts off at 100%. So if that particular generation, we've got time in years and on the x-axis there, if that particular generation becomes exposed to something that damages the microbiome, so you'll get a drop off here, and I don't know if you can see my cursor, but you can see the drop off looks like steps. And that might be mitigated by the fact that you could pick microbes up from the environment that kind of mitigate that, and that's what you see with the dashed lines there. So this horizontal acquisition. But that is not a very effective process in comparison, because we know that these microbes are more easily integrated into your microbiome in that window of time early in life, maybe not so easily acquired later in life. And so if you keep looking over generations, if you have generations that are constantly exposed to these kinds of detrimental lifestyles, then eventually what you'll see is a drop-off in diversity that cannot be mitigated by horizontal acquisition of microbes from the environment very easily. And when Blazer and Falco sounded the alarm on this, they were sort of suggesting that this might be one of the most important things that is contributing to the increase that we're seeing in chronic diseases in the Western world right now. So it's not just pharmaceutical drugs, as I mentioned, it's also the Western diet. The Western diet is particularly bad compared to some. We know that the average Western diet is very rich in refined foods, it's low in fermented foods and complex carbohydrates and fiber. Refined foods are easily broken down in the upper GI tract. And so what that means is that they feed the human part of you very, very well, because that's where you absorb a lot of the sugars, a lot of the energy, but there's very little leftover food that makes it into the colon under those conditions. And most of your gut microbiota diversity is actually present in the colon. Colon's a very special organ, as you'll see in a little while, sort of a specialist organ for keeping your microbes safe. When you starve this microbial community, it can lead to ecosystem damage. You know, there's only so much you can live without food, and the same is true for microbes. Eventually, they will die off, they will become extinct from that ecosystem, and you'll end up with reduced diversity. So we do know from research in the literature, two very seminal papers here, but there have been many others now that are also exposing several other food additives as very dangerous for the gut microbiome. The two big ones are artificial sweeteners, particularly sucralose and aspartame, and artificial food emulsifiers. These are the things like polysorbate and carboxymethyl cellulose that are added to a lot of sort of foods to keep them in sort of shelf-ready form, so they don't look horrible. They don't separate the oils from the aqueous phase from the oil phase, and they're used a lot in sort of especially in cheaper foods. Not all emulsifiers are bad, but the artificial ones, the cheaper ones, seem to be really quite dangerous to the gut microbiome. They damage the gut microbiota in ways that, again, I don't have time to explain in too much detail, but basically what we see is a loss of diversity and also a sort of a bias towards microbes which are more inflammatory within that ecosystem. So you lose microbes and you also gain in complex abundance, if you like, microbes which are not particularly friendly to the gut, and that can lead to inflammation, and it kind of sets up a sort of a vicious circle of inflammation that can lead to more loss of diversity and so on. So it's really not a good situation to be in. And of course, I couldn't go without mentioning the fact that antibiotics, of course, are also very clearly damaging to the gut microbiome. Antibiotics are very, very important in medicine, and so I'm not saying that we shouldn't take them, but I think that we have had a situation where in the past they were taken rather a lot because we didn't appreciate the fact that they are not just, they don't just have an action on the pathogen that you're trying to rid yourself, on the infection that you're trying to get rid of. They have very broad scope, if you like, to damage microbes, and those microbes in our bodies as well. And so there's been many, many studies, and even since I made this slide, many more that show that your gut microbiota changes very significantly with antibiotic use. It takes a long time afterwards to return to baseline, although it's different for every individual, because remember, every individual has a different microbiome. So we have no way right now of predicting who is going to suffer more from an antibiotic exposure than someone else. And when you have repeated hits, in other words, if you're given an antibiotic that doesn't work, so you're given another antibiotic perhaps to try to get around any resistance issues, and maybe that's a different class of antibiotics. What you're doing is you're basically giving your microbiome multiple hits, so it never really gets a chance to recover, and that's been very clearly shown in the literature as well. But it's not just antibiotics. Well, what about other common drug products? So it's really interesting, actually. The question in pharmaceutical science for a while has been, why do some drugs work miracles in some patients and they don't do anything for other people? And that's been a big problem. No one really understood. There's a lot of money being spent to try to show it's to do with the human genome, it's to do with your genetics. But actually, the issue is that we have been focusing on the wrong genome. So your gut microbiome is another genome, right? It's another part of you, and it's different for every single individual because you have a different microbiome. And your gut microbiome carries out a great deal of metabolic work for you. So if you're damaging that gut microbiome, you can see how, or you're changing that gut microbiome, you can see how you might have a different response. So predicting the drug effects of whether something's going to work or not is really not an exact science right now. In fact, there is a science that is emerging, a new era of pharmacology, and it's called metagenomic toxicology. And I think that that's going to be a really interesting field to watch in the coming years, because starting to understand which microbes you have, and even augmenting your microbiome with drugs that might, with microbes that might change drugs or help to boost a drug's effectiveness in some ways, that's already becoming a reality. And it's a very exciting area of research. And just to sort of show you again, this is the study from that proved to us that Blazer and Falco's thesis was actually quite astute. This is a study from a couple of years ago now, but what they did here is they looked at the population of immigrants coming from Southeast Asia over to the United States, and then looking at them from the point of view of their gut microbiome as well as their BMI. Knowing that in the United States there's a big problem with obesity right now, they were asking the question, and knowing as well that obesity is very closely related to the makeup of the gut microbiome, they asked the question whether or not these immigrants were going to be at risk of obesity, and whether or not that might be connected to their gut microbiome diversity. And what you can see is that these immigrants, when they first come across the water and settle, their microbiome, you can see changes. They're very subtle, but as time goes on, and they've been there for a long term, or the next generation from them has come about, you can see that the gut microbiome in these individuals is in fact much less diverse than the original. And it's sort of in line with what we see with European American settlers who have obviously been there for a lot longer. And so the idea here is that again, that loss of diversity is leading to chronic diseases and in this case, obesity. So what we need to understand is that the safety of lots of food additives and supplements and drugs and things like that have never been assessed taking into account our microbial passages. And so it's a really new area of research, a really important area of research that we need to look at. So we're just scratching the tip of the iceberg. And I think it's a little bit frightening, actually, for many, certainly for me, that we just know so little and we do so much to ourselves, to, you know, in terms of our lifestyles, without really understanding what we could be doing, you know, any detriment to our gut microbiome. And so to sort of introduce to the next section of my talk, so there are lots of diseases now that have been associated with this altered gut microbiota diversity. And the more we look, the more we see. And again, I could add far more things to this slide, but I just wanted to point out a few things. So now, of course, this is association, all right? So association is not the same as cause, as we know, and some of these diseases, the very kind of symptoms of the disease may contribute to a lack of diversity as well. But what we do know is that for some of these diseases, there absolutely is a causative or a causal relationship. And the two that I want to pull up are, of course, C. difficile infection. We know that that is only caused or only happens when we have a situation where a gut microbiome is damaged, usually through antibiotic use. And as a result, the C. difficile overgrows and causes a problem. And the other one that's interesting and emerging is colorectal cancer. And that one I've been working on for a number of years now. And what we've discovered there is that there are issues with diversity in the gut that change the composition, if you like, of the gut microbiome, so that less abundant microbes that could be problematic now sort of come up in numbers and start to cause problems. And in fact, a small group of microbes, including one of my favorite microbes that I've been working on since I started my independent career, which is called Fusobacterium nucleatum, is what we call an oncomicrobe. It's actually, we discovered that it causes cancer in a little bit like Helicobacter pylori, but in the gut system. And there are other oncomicrobes as well that are being discovered all the time. So we do know that that is the reason that those microbes are there and are allowed to proliferate again is because of a lack of diversity. So that's another interesting area. And for all of these other things, there's certainly a lot of evidence that that a lack of diversity is somehow contributing to these diseases. It's very difficult to prove causation. It's far easier to prove association. So the studies that have been done are pointing in that direction, but I think more needs to be done to see what exactly is going on. And this sort of brings me to this other idea. So how do we actually know that Westerners have low gut diversity, right? Because most people that have studied this have been looking in the last 20 years or so at Westerners, very, more often than not, these are studies that are done in the Western world. So the study subjects they have are Westerners. So we can't go back in time, obviously, to look at microbiomes pre-antibiotics and refined foods, although a little bit of work has been done, interestingly enough, looking at coprolites from cavemen, from early humans, if you like, to see what sort of microbial diversity they have. That's actually been very difficult, as you imagine. That ancient DNA is very difficult to do much with. So it's not very equivalent or not very comparative. But what we can do is we can look at indigenous peoples who have not had exposure to the kinds of lifestyles that the Western world has had. And there's sort of very famously the Hadza tribes people in Tanzania have been studied from this point of view, as well as the Tunapuco people in Peru. And in fact, my own research for my Canada Research Chair, we were very interested in a group of Yanomami people in the Venezuelan Amazon who have, through, again, and it will be another story for another day, but we've managed to actually procure stool samples from these villages in this very remote area of the jungle that takes 3 days to trek to. And we have those back in the lab, probably the most precious things that I've ever had in my lab, precious samples. And we're starting to actually culture these to actually look at the microbes that are in them and finding all sorts of things that we have never seen in the Western gut microbiome. The importance of culture, of course, is so that we can actually understand the physiology of these microbes and start to try to understand what they're doing in these indigenous people, whether or not they are beneficial or not, or part of an ecosystem or something that we've lost. But again, without sort of spending too much time on this, the amount of data that we have already shows that it's not that these people just have picked up different microbes from the environment in which they live. Many of the microbes that they have look like they are ancestral microbes that they have had, since the early time of Homo sapiens, that we have actually lost through extinction events. And the reason for that is because, you can look globally at these people in South America and in Africa, and they have the same microbial species that we Westerners don't have, and yet they're separated globally by huge distances. Now, the other thing that we are dealing with is that we have this kind of pervasive advertising message in our lives right now that all microbes are bad. And it's a real shame because, of course, all microbes are not bad. There's all of this sort of antimicrobial stuff going around. And this is compounded right now because of the COVID outbreak, because, of course, we do need to be using hand sanitizer, and we do need to be sort of mindful of the fact that that we could pick up viruses. But the problem is that when we have this perception that we need to treat everything with antimicrobial substances to keep everything super clean, unfortunately, what it's not telling us is that only a tiny fraction of microbes are pathogens. And most of the microbes that we have on our bodies or in our bodies, in fact, the vast majority are actually beneficial, or at the very worst, they are just neutral. Now, antimicrobial practices don't discriminate between pathogens and microbiome. What I've added here, though, is it's kind of interesting. So as well as we have all this hand sanitizer around at the moment, but companies that used to be making hand sanitizer have started to capitalize on this idea that the microbiota, especially of the skin in this case, is actually quite important. And I've actually started making products that have skin microbiome impregnated in them. So it's kind of an interesting paradigm. I think that this is sort of halted for now because, again, of COVID, but I think that that this will become an important industry in the future. We're also starting to use our knowledge of microbiome composition to predict health. And so this is a paper that came out last month. As I said, lots of papers at the moment. It's kind of overwhelming. This one is talking about something that we call the Gut Microbiome Health Index. It's a new mathematical formula for predicting the likelihood of disease from your taxonomic signature. And it's something that was previously very, very difficult to do. This paper, if you're a mathematician, I recommend you read it. It's very, very dense in terms of mathematics, of course, as it should be, looking at huge data sets of people to try to associate disease with composition. And in fact, there are some trends. And so it's interesting that we are starting to get to the point where we can make predictions about people's health based on their gut microbiome. So that brings me to the last part of my talk, which is this question: what can we do to support gut microbiome health? So there's a number of activities that seem to support a healthy gut microbiota. One of the most important ones seems to be natural childbirth and breastfeeding. Those seem to be extremely important. So I think we've perhaps known that for a while, but I think especially, in my lab, we were very interested in human milk oligosaccharides, which are very special sugars from breast milk, which seem to be extremely selective for the support of certain microbes in the gut, and seem to play a very important role in actually assembling the right microbiome in an infant's gut. Those things are not present in formula milk. I think Nestle has come out with a new formula milk, it's very expensive, which contains one human milk oligosaccharide, the cheapest one, the easiest one to make. But human milk oligosaccharides, there are around 60 of them in the average person's breast milk. They're different from person to person because it depends on your secretor status. But I think that what we're beginning to understand is that that breast milk and those human milk oligosaccharides, from the point of view of the microbiome, are far more important than we ever realized. And so that's a very important thing. This is a tricky one because of the current times. Very difficult to have a lot of social exposure right now. But having increased social exposure actually does increase your gut microbial diversity, and it's probably most important again when you're a small child. And so I do worry actually about these current times and whether or not we'll see an impact of this in years to come. But I guess we'll find out. This is an interesting one. Regular exercise has been shown to be extremely important to support a diverse gut microbiome, and we don't really know the mechanism for this. But it does gets me up and on my treadmill every morning, because I've seen the data and I believe it. It's a very interesting one. And interestingly enough, the effect is seen more in women than in men. And but again, we've seen in both sexes, but more in women than men. We don't know why. Outdoor activities, you can probably imagine that when you're outdoors, you have more exposure to microbes in the environment. This one is good, pet ownership. If you have a dog or a cat or some other furred animal that goes outside and brings microbes to you, then that's a kind of a lazy way of getting your microbes, I guess. But that's been shown. So pet ownership is definitely associated with a higher gut microbial diversity. A healthy diet, that's a big one. And antibiotic and other drug use only as appropriate. So in other words, it's not good to stop taking antibiotics just because you think they're bad for you, because in fact they can save your life and they can actually help your health if they're used appropriately. But when they're not used appropriately, or when they're used probably less judiciously than they should be, then they can obviously be problematic. And these are all most effective if adopted in early in life, although they all have an effect, or most of them have an effect later in life as well. And this is just, again, this paper came out last week. And so I put it up because, again, there's a flood of information that's coming through that's only possible to do now because we have these huge data sets and the mathematics and the computational ability to look through them. So here we looked the group looked at thousands of different people. Now I know that the graph on the right, there's lots of tiny words. I don't expect you to see them. But I encourage you, if you can, to look this paper up if you're interested in this at all. What they did is they took a whole bunch of different lifestyle and anthropomorphic and other kinds of markers, if you like, of health or disease, and they correlated that to diversity. So this is a left C plot. So what you'll see on the sort of top half of the plot is a sort of a negative correlation. So in other words, this means that the situations on the left-hand side and on the y-axis there are correlated with less diversity. So you can see the top one there is the LPIR score, which is to do with type 2 metabolic syndrome, type 2 diabetes. So all of those things. So these are associated with less diversity. On the flip side, on the bottom side of the bottom half of the graph, you can see the things which are correlated with high diversity. And so the one that's really surprising to me, and again, I suggest you dive into this data because it's fascinating work, is that height. So in fact, if you're tall, you're actually more likely to have high microbial diversity in your gut, which is really interesting because I'm short, so I kind of love. I lucked out. I didn't luck out there, but really, you know, something that was very surprising even to the authors of this paper, something that hadn't been considered that could be quite important. But these kinds of associations will help us to understand how we can start to manipulate the gut microbiome in the future in terms of for health potentially. So then how can we fix dysfunctional gut microbial ecosystems? So if you have an ecosystem that isn't very diverse and it's not working very well, can you fix them? So can you fix them with probiotics is the first question. So you might have been told if you take an antibiotic, your GP might have even told you this, and I try and educate them not to say this, but you might have been told that you can just cancel the negative effects out of an antibiotic out by taking a probiotic. And the answer to that is no, you can't. Unfortunately, there are many different types and strains of probiotics to start with. There are many manufacturers, some are legitimate and most of them are not. Many overinflated claims, like massively overinflated claims, very little actual science. And there was a study, this is one of several studies, but this one is particularly interesting. 14 commercial probiotic products, they took a look at what was on the label and they took a look what was actually in the capsules. And they found that for the most part, most of them didn't have what they advertised to have in the capsules, and in some cases they actually had some potentially pathogenic organisms in there as well, which is really not a very good thing to have. Now, the layperson's view of probiotics is something like this: that you can kind of go into the supermarket, into the dairy section, and there's a whole bunch of like kefir and yogurt and all sorts of things that you can take that's going to enhance your gut microbiome. And I think that several manufacturers, no names mentioned, they capitalize on that. Now, you'd have to eat an incredible amount of yogurt to actually get to the numbers of microbes that that might even have a hope to get into the colon to do any kind of potential beneficial things. So the first myth, of course, is that the probiotics found in food are the same kinds of species that are found in the gut. That's not true. The probiotics that you found naturally in food that ferment yogurt, the milk into yogurt, those are quite different genetically from even the same species that might be present in the human gut. The ones that are present in the human gut have a different genetic repertoire to allow them to live in that you know, fairly harsh conditions. And the second myth is that probiotics colonize the gut. They don't. So there have been a few studies that have shown that under some extreme circumstances, you might see them hanging around for a couple of weeks or more. But for the most part, they are just going to pass through your gut, and they are not going to stay, they're not going to colonize, they're not going to stay with you for any period of time. And when I think of probiotics as a microbial ecologist, I have a kind of a skeptical view, which is this. So your normal gut microbiota, as I mentioned before, in your colon, you've got around 100 billion to a trillion colony forming units per mil. And in a probiotic capsule, your average probiotic capsule, you've got 2 to 15 billion CFU per capsule. Okay, so that is a drop in the ocean. And you have to remember that that capsule has to get through your gut to start with to be able to get to the colon where it's going to have it's the most benefit. Now, that's not to say that all probiotics are bad. I'm just trying to point out that many of them kind of, they don't fit because they're marketed as food products. They're not drugs at all. So the bar to pass as a food product is very low, and the claims that they can make seem to be less well policed. So for example, strain is important, not just species. So probiotic species, there's no such thing. A probiotic strain, there is. Okay, so you can't just call something a probiotic because it happens to be in a species with something else that is a probiotic. That doesn't work that way. So on the left-hand side, you can see Culturelle as an example. There's clinical evidence for the efficacy of this particular probiotic. They have done some clinical trials. The probiotic species and its strain are very clearly labeled. And then on the right-hand side, and again, I'm you know, perhaps unfairly pulling up this company for making this, but it's sort of symptomatic of other generics. They use the same kind of colors in their boxes, the same kinds of messaging. And in this particular one, there is no clinical evidence for efficacy. And when you look at the box, the species name is not given. Okay, so the probiotic species, the species name is given, no strain is given on the box. And you have to be realistic when you're thinking about probiotics as well. So you wouldn't normally expect that a drug designed to treat diarrhea would also work to treat psoriasis, for example, would you? But unfortunately, we tend to think of probiotics as panaceas that are going to do everything. They're going to be amazing for our health in every way. The benefits that have been seen in clinical trials are modest at best. And that's not to say that they aren't there. For some of the clinical trials, it's been quite impressive what these things can do. The real benefit of probiotics, the good ones, is that the risk of side effects is extremely low. They're very well tolerated, and so it's worth a try if you pick the right probiotic. So the next question, of course, is how you pick the right probiotic. Well, luckily for us in Canada, we have this great resource called the probiotic chart. This is a clinical guide to probiotic products available in Canada. If you go to this website, you can see all the different probiotics that have had clinical trials associated with them and the effectiveness of those clinical trials for given indications. And I think that's the right way to start looking at probiotics rather than, you know, all of these things you can buy in a health food store. So this is a really good resource. I really recommend it. So what about fecal transplants then? So if probiotics are no good, what about fecal transplants? So this is pretty unsavory. You use a healthy donor's microbes to replace your own, and it's very crude. Generally, you take a stool, you put it in a blender, and you instill the homogenate into a patient through an enema or colonoscopy or a nasoduodenal tube. That's how they do it in Europe. Or through capsules, which are just frozen encapsulated stool or freeze-dried stool, which is being done in Calgary. Now, for C. difficile infection, this results in the cure of the patient in over 90% of cases, which is really impressive. I don't think there are many drugs on the market, if any, that have that kind of rate of efficacy. You get a rapid resolution of disease. You only get a rare recurrence of infection, but there are many, many risks. You're taking a drug that's essentially a waste product. or you're taking a waste product and you're using it as a drug. And there are many, many risks associated with the unknown microbes. You can screen the stool for everything that you know about, but we can't screen stool for things that we don't know about. So especially when we're talking about in terms of colorectal cancer and the microbes that we are discovering that seem to have a role in that take years to have their effect, the risk here might be a little bit more than is considered. And so you really have to do this risk benefit. And so that kind of leads me to the very last part of my talk, which is what if we could make fecal transplant, fecal microbial transplant, FMT, what if we could make it safer? So couldn't we just select some pure microbes from healthy stool to create a microbial ecosystem therapeutic? Well, in fact, we did. And so again, I don't have too much time left to talk about this. And I've taken a few slides out because it's becoming a huge part of my life right now with New Biota that we founded in 2013. But what we've done is we've created a product which we call MET2. In fact, we have several products now as well on top of this. But MET2 is a new first-in-class biologic drug, which what we did is we took a lyophilized standardized ecosystem of microbes and we encapsulate it for oral delivery. And we have clinical trials. We have one that's actually now finished for recurrent C. difficile infection, and the paper is in review. So I won't reveal too much, but it was a successful trial, and several other indications. So we have one for an oncology trial. We have depression, anxiety one, and we have metabolic syndrome, and several others coming through the pipeline. Now, this will not be a probiotic. It will be prescription only. So just to give you an idea of what some of the things that we've had to think about, the commercial and patient considerations. So we have to think about the needs of the ecosystem. So stability, diversity, functionality, strain interaction, all the things I do in my lab, in my academic lab. But we also now have to think about the needs related to commercialization. And those are quite considerable. And of course, if you want to make a business work, you have to really consider these. So we have a few things that we have to consider there. And of course, we've considered the needs of the patient. Now, in the past, when we first sort of started looking at this sort of microbial ecosystem therapeutics, we were thinking in terms of just shooting a suspension into the colon directly, but patients don't like that, understandably. So we've actually done the work to make this into a lyophilized product that you would take orally, just like you would take a probiotic. The sweet spot seems to be around 30 to 40 different bacterial species. Excuse me, at the moment, as long as you keep that phylogenetic diversity. Again, it's about keeping that diversity of biochemistry, if you like, to make sure it's there. So how do we determine the needs of the ecosystem? Well, as I mentioned, the human colon is a very specialized organ. It's a sophisticated bioreactor. So in my lab, all we've done is we've recreated it just with glass and stainless steel into a chemostat bioreactor. So we now have chemostat bioreactors in the lab that allow us to look in vitro at the microbes. It supports the microbes in an environment similar to the gut. It's a very, very useful system for understanding microbial ecosystem function and dynamics, and it's really allowed us to take our microbes that we isolate from a given donor to create these microbial ecosystem therapeutic products and to be able to predict how they might work in the gut environment. And how do we standardize these live microbial therapeutics? Well, this is kind of an ongoing trial. So these are biologic drug products. They're not probiotics, as I mentioned. There's a much, much higher barrier to approval for this kind of product. Each individual isolated component, our 40 species ecosystem, is considered a drug product. So in fact, we do quality control on 40 different drugs to make our end product. It's incredibly time consuming. But I feel like we're taking the high road here. We really need to do this because we need something that's safe and controlled. And so we've done an awful lot of work on microbial physiology. Growth rates for unpredictable microbes need to be predicted. So we've had to do that work. And we've also done an awful lot of work to develop novel methods novel approaches to quality control, because the current methods used by Health Canada and the FDA require very set protocols that they're very reluctant to leave. And so we're trying to fit new molecular methods to this kind of procedure. And our products don't quite fit the box, so we'll see how that goes. We're doing a lot of work on that, lots and lots of reporting and lots of review panels, and but I think we're making some headway. So in summary, hopefully from this talk you recognize, or you see the microbiota 3 R's. The first one is to recognize that we're custodians of a hidden army of helpful microbes. The second is that you need to respect what they do for us. And the third is that you should reinforce their beneficial activities. And then when things go awry, we think that we might be able to have these new approaches in medicine to help sort of improve the gut microbiome diversity, which might be helpful for benefiting health. So with that, I just want to acknowledge the people in my lab. This is not a terribly new photo, unfortunately, because if I had a photo of everyone now, we won't be wearing face masks and you wouldn't be able to see them. So this is most of them. And sort of separated that they tend to work together right now, New Biota and my academic lab. So I think they benefit from each other. So it's a really great working environment. And I also want to acknowledge all the people that fund me. They're down at the bottom. And on that note, I thank you for your attention. I know we have a break now, and then I'll come back for questions afterwards.
Okay.